Dr. Joel Dudley, the director for the Institute for Next Generation Healthcare, will be giving our State of the Science address now. He's made his way through Manhattan traffic.
Presentation on here. But since Dr. Horowitz mentioned several times I'll be talking about Alzheimer's, but in fact I will not be talking about Alzheimer's. The reason being is we're hosting this conference. This is not a platform to talk about my research. This is I can just give you a copy of the right presentation.
Okay.
All right. It's a platform to talk about all the great research that's been done by all the other collaborators in the initiative here. But if you are interested in the Alzheimer's work, I'm happy to talk about it. I think the key finding from that Alzheimer's study, which Dr. Horowitz mentioned, which was published in Neuron earlier this year, was that it was using sort of a big data approach to analyze which was the largest, probably Alzheimer's multi-genome omics data set probably available now. And using a data-driven approach, we were able to identify HHV6, herpes virus, actually HSV1 and also HHV6 as a potential driver of Alzheimer's disease, and not only found an association, but mapped out specific molecular pathways. And actually what we found matches quite a bit of what Dr. Horowitz was putting in his theory. What was nice about that study is we brought a lot of the specific molecular pathways and things like that we can actually test. So this is how the medicine can complement, or the science can complement the medicine. I think I bought myself enough time. And here we go. All right. And I want to thank Dr. Horowitz for speaking for me, since the travel gods tried to thwart me in every step to get here today. Right. Hey, see the science. Look at that. All right, I got it.
Yep.
All right, thank you, Melissa. And by the way, the real story there with Alzheimer's is that, as was mentioned previously, is that this real story is not about these particular viruses, HHV6. It's about innate immunity, and the activation of innate immunity is really the key driver to Alzheimer's. We're not the only ones that believe that, which is really interesting because it springs a link to, in fact, Lyme disease and the infections that come with Lyme disease. All right, so thanks for coming here. I know it's a big change of venue. It can sometimes throw people off, but I love this venue because I feel really powerful when I talk with you there and all the background there. I feel like I'm a master of the universe here, so it's really fun. So before I get into the science, though, I want to talk about, so I'm sort of speaking to, I think, patients and people who are not scientists. This is sort of the goal of this talk, to just remind people that biology is really, really hard. And so there's rarely smoking guns in human health. I always use this picture. And what do I mean by that? There's rarely the one magic gene, the magic protein. Am I on screen? No, I'm not on screen. So rarely smoking guns in human health, what do I mean is there's rarely the magic gene, the magic drug target that's going to solve all of our problems for the most part. A great example is I have inflammatory bowel disease, so genome-wide association studies on inflammatory bowel disease have found over 200 genomic loci associated with inflammatory bowel disease. Think of that as 200 genes associated with Crohn's disease, roughly. By the way, those 200 genes don't even explain half of the genetic component of that disease so far. So we're 200 genes in. We haven't even explained half of the genetics of that particular disease. Where's the drug target in that? So this is the case for all diseases, for the most part, common diseases, diabetes, Alzheimer's, et cetera. And of course, Lyme is going to be the same thing, right? So really, what we're dealing with is a complex system, lots and lots of different factors. We're probably unlikely going to find a magic bullet. And I also like to point out, just to get people to set expectations just on science in general, is that it's really hard to go from knowledge to a solution or to understanding. So this is actually a review article from Nature Reviews Genetics on cystic fibrosis. So cystic fibrosis, you may be aware, is a rare inherited disease where the mother and the father have to pass on a mutation in a CFTR gene to give cystic fibrosis. It's fatal, in late teens, early 20s, or some people are living longer now due to some drugs. But the point being is that here's a case where all of essentially the prevalence of cystic fibrosis is explained by mutations in this one gene. Now there's lots of different mutations, but this mutations in this one gene, CFTR. So we have this case where we have one gene, one disease, right? And we know that this gene is the single causal genetic driver of cystic fibrosis. And yes, actually, we've known that for 20 years at this point, right? So and we actually don't have a cure for cystic fibrosis, so this is pretty, we should be humbled. And I just want everybody to appreciate, while we're making great scientific strides, the science is really hard. Here's a case where you think one gene, one disease, 20 years later, we're still struggling. But it shouldn't be surprising that it's so difficult to treat these diseases, even when one gene, one disease case. Why? It's because the way human physiology is wired, we have these sort of complex layers of networks, if you will. If you think about a single cell in the body just for 1 second, you have single cell, we have 3 billion base pairs of DNA, as you probably learned in high school, wrapped in a three-dimensional conformation of various chromatin states, wrapped around histones like a spool and thread, right? 3 billion base pairs, and they express, you probably learned in high school biology, RNA. You probably learned mRNA, but actually they express a whole universe of RNAs, link RNAs, picoRNAs, eRNAs. The list of RNAs goes on and on and on. So there's a whole, these go back and bind to the DNA, regulate the DNA. Some of these RNAs, like mRNA, make proteins, right? But proteins then form complexes. They interact with one another. They go back and bind to the DNA, change the regulation of the DNA. We haven't got out of one cell yet, right? So there's a whole constellation of millions of interactions in a single cell. Cells form tissues. Tissues have cell-to-cell communication. Tissues form organs. We have organ-to-organ communication. And if that's all we had inside this box, life would be easier, at least. But we have the pesky environment interacting, especially in the case of Lyme disease, from all sides, right? So this is the challenge. I just want to sort of help people understand why we have these great scientific discoveries, that it's just really, really hard. But luckily, we got a lot of really good people on the case here in Lyme. So what I'm really going to do here, again, this is not to talk about my research. I'm going to just highlight some papers that came out in the past a year, roughly or so, and sort of explain why I think they're important, why you should know about them, and pump up other people's work for them, so they don't have to toot their own horns. So, and a lot of these papers come from folks in this consortia. So the first paper I want to highlight was a paper on actually this comes from ISB, right? And where's the ISB, folks? I believe this paper comes from ISB, yeah. So what they did is they used next generation sequencing technology to sequence Lyme strains in the wild, right, from animals. And using next generation sequencing technology, so genome sequencing technologies, this is the novelty here. And they found, of course, lots of different strains. Now, the key challenge here is that we don't have lots of Lyme genomes. I don't know if you know that. So we have lots of human genomes, right? We're getting more and more. And the variation in the Lyme genomes is a really important thing, because as there's variation in Borrelia, right, that changes the virulence factors, right? But we don't have really a big sort of genomic map of all the variation that can be found in nature in Borrelia, right? We just haven't done whole genome sequencing of enough of these bugs. And even small changes can be really important in the virulence. So for example, there's a paper published by some Mount Sinai colleagues a couple of years ago on E. coli, and an E. coli strain that became virulent in Germany and contaminated the food chain. It's actually a very, very small little change in that genome that made it super virulent, from benign to virulent. So that's why we need these maps. One thing that's really, oh, this isn't the ISB study. Look at that, I'm screwed up right from the beginning. So these guys, this is not the ISB study. ISB study is coming next, but this is an NGS story anyway. So in this case, this is what's really useful to take away from this study is there's a way for culture-free identification of novel Lyme strains using an NGS approach. And of course, a lot of these bugs are hard to culture. This is the power of that. This is, I'm going to try to pick a money shot sort of figure from each study here. And this is one that where they selected various samples. And this is just looking at OspC, an important gene obviously for Borrelia virulence, and all they're showing you here is using sequencing. They're able to identify novel strains, et cetera. This is the ISB study. I apologize to my ISB colleagues. Continuing the NGS theme, this is where they addressed the issue in this case that there are only 47, sequence data on 47 strains of this particular strain of Borrelia, which is important. So we need to build out this bigger data set. One thing that's really important is that in this study they used something called long-read sequencing, Pacific Biosciences sequencing. So if you don't know how current genome sequencing technology works, you kind of sequence little pieces of the whole genome, like little puzzle pieces, right? And then you put them together, But the problem is when you have these puzzle pieces, you got to create a reference. You have to, you need the picture on the box, right, of the puzzle pieces to sort of figure out how the puzzle goes together. Well, that's called a reference genome, that picture on the box of the puzzle pieces. So you can only sort of assemble the genome for if you have another genome that looks similar, right? But if you have totally novel genomes that you can find, you need really, really, really big puzzle pieces, right? So what they did, one thing important that they did in this study is they applied Pacific Biosciences, which is on sort of third generation sequencing technology that gives us ultra long sequencing reads. That lets us do de novo assembly, which means we can assemble the genome of the novel species we might find in a sample, and without using a reference. So the point being is this is sort of the first application that I'm aware of using PacBio sequencing, and maybe I could be wrong. And also, if there's any study authors in here that I'm totally misrepresenting your study, feel free to raise your hand or call out something you think is more important to cover than what I covered. But again, so then they used the PacBio sequencing technology and released a reference genome, a de novo assembly. So, and all they're showing in this particular case is comparing two strains, and what you find using this de novo assembly technique is a lot more changes between these two strains, some probably important for virulence. You would not have been able to find a lot of these if you had used the traditional reference genome assembly. So the takeaway is we should use more PacBio sequencing to discover novel species, right? So another interesting study that came out was, this is also part of the consortia study, I believe, on a tick-borne disease, Serochip, right? That was designed to discriminate between 8 tick-borne pathogens, which are listed there. You're able to read yourself. And really the takeaway is that this is really a promising new tool for diagnostics. One of the things I really liked about this paper is not only they gave their antigens for detecting, because they did a lot of really painstaking work to figure out what were the right antigens, and they actually provide the peptide sequences, that they're targeting against to detect these antigens to develop this diagnostic platform. This is an interesting paper. Rick talked about this a bit. Again, there's a different crowd here today than was yesterday, so we want to make sure people are aware of it. Designing a large-scale study looking at hundreds of neighbors and neighborhood and hundreds of actual neighbors in New York. One of the most important things about this study that you should be aware of is it's probably, in my opinion, one of the most thoughtful and rigorous studies of the sort of Lyme envirome to date, at least in conception. So we'll be able to answer really important questions about the environment effects on Lyme that we weren't able to answer before. I couldn't show you a cool figure from this paper, though, because I couldn't access it. So hopefully we can solve that. I got some commentary on that at the end here. This study by Dr. Salati here was a tour de force. I wish I hadn't read this study because it was so good, and I spent most of my time reading this instead of doing the other things I was supposed to do. But the novelty of this study was to identifying novel immune targets in Lyme. So that they used a mouse study to looking at cyclic AMP enhancing drugs to increase IL-10 production, which can counteract some of the negative inflammatory effects of Lyme infection. And what they really did was attack or target the host immune process, right? So this is the novelty here. And Tim, feel free to correct me if I'm wrong. Yeah, so targeting the host response, right? So obviously we use antibiotics to target the bug itself, but this was a really interesting, almost immunotherapy, if you will, right, for treating the inflammatory response to Lyme. And in a way, it was actually, I do lots of drug repurposing, personally, finding uses for existing drugs. So this was effectively drug repurposing, for immunotherapy in Lyme, where we're addressing the immune response of the host. It was a really amazing study. There were too many slides in here to pick just one that were really good. He really did a good job of teasing apart the biology. But the one thing that was notable from the study is actually this animal model that I hadn't been aware of this ankle thickness model, which we've talked about with some other folks lately. So this was the first, at least, study that I was aware of that had a compelling use of this model. So a novel method to integrate host factors. So Dr. Embers talked about this paper a little bit yesterday, but again, for those that weren't here yesterday, I think it's really important to be aware of this particular study. And by the way, if I don't mention your paper, no hard feelings. I only have a limited amount of time. this isn't a rank order of my favorite people in the consortium or anything. So it's totally random. So no, so what they did is used a rhesus macaque model and infected them with Lyme, and also looked at post-treatment. And the goal of this study was to look at sort of the presence or distribution of Lyme across physiology. So there's two really important things. Is that, from my understanding, this is the first study in primates to examine pathological findings of persistent Borrelia in the late stage of Lyme disease after treatment. And that obviously this model has very promising benefits for studying Lyme in humans as sort of a novel approach to understanding Lyme in primates. So this is one of the papers with so many cool pictures I couldn't pick one out, but I liked the D slide there, which if I remember correctly was actually post-treatment was the presence of Borrelia in a nerve, right, in the nervous system, right? Is that correct? Which one? Huh? All right. At least it was a nerve. That's great. Actually, I could tell you it was anything, to be honest. Yeah, you wouldn't know. Right. Yeah, I'm just kidding. Right. No, it was a nerve. No, so it's pretty amazing. So this is one thing we actually don't know. We talked about this yesterday with some of the speakers. Is we really don't know. I guess someone's funding this post autopsy or post infection. I mean, we can't do this to live people. We really don't know a lot about the sort of tissue or physiological distribution of the bug in not only the active state but potentially the persistent state. And this is going to be an amazing model to tease that apart. This study was talked about quite a bit yesterday, but again, for the folks that were not at yesterday's meeting, I want to highlight this study again because it's one of my favorites that I've seen today. It's a rigorous study of food as medicine. So you can go out on the books or the shelves of Costco and find all kinds of books of food as medicine. But the problem is there's not very many rigorous studies that have been done on sort of food as medicine, and so it's hard to convince mainstream researchers, doctors, et cetera, that it's a viable approach. So I love this paper because this is a very thoughtful, sort of rigorous scientific study of food as medicine, where they're looking at some essential oils and their activity against Borrelia, comparing it against known antibiotics, and oregano and cinnamon bark and a few others come out as some of the top compounds for having activity against Borrelia, and also evaluated some ability to bust through the biofilm, which nobody likes biofilm. But the one figure I chose out of this paper, there's many cool figures in this paper, was that so one thing when you evaluate drug studies, for anything that's like a drug study, is you have to look at the concentration at which something is active, right? So a lot of times, if it's not at a physiological or low, concentration, then it's not interesting because the drug will never be at that high concentration in someone's body. So I think one thing that was really amazing in carvacrol is, which is a component of oregano, was active against both stationary and active Borrelia at very, very, very low concentrations. So is that correct? All right, all right. He's even filming it, so I must be doing a good job. So this is pretty amazing. And with all of these, food as medicine studies, it's always hard. So we've actually, on the drug discovery side, looked at a lot of natural products. One of the really hard things with natural products is to tease apart the component that is contributing to the biological effect that we might know exists with food. So this is also a great job to drill down into the specific component that we could isolate that might be explaining the effect of oregano. So since hopefully there's some patients here and not just a bunch of scientists and others, one of the things I wanted to talk about as we talked about some of this research is, as you noticed, I couldn't even get some of the papers we talked about. One of the big challenges in evaluating the science of Lyme disease is we just can't even get access to the studies, right? So you may not be aware of something I shouldn't be talking about, which is called SciHub. So SciHub was something that I think was started in Kazakhstan to liberate all of the published studies. So we have a very broken publication system in modern science, where we do a bunch of work, and then we pay people to publish our papers, and then we turn around and we pay for them to access our own papers, which is a pretty amazing business if you can get away with it, for sure. So if you can't get access to a paper through conventional means, and if you exhausted all of the normal routes to get access to a paper, there's something called SciHub, which has all of the published papers in it. I can't tell you where it is. As a researcher who publishes in mainstream journals, I shouldn't link to it, but I can link to the Wikipedia article about it. So if you're curious about SciHub, and if you're a patient or if you're a researcher that's not at an institution like Mount Sinai that pays like $1 gazillion a year to access all these studies, there's a little something called SciHub you can read about for informational purposes only, and get access to papers. I think this is really important because, again, I could only cover a small number of papers. It's a huge challenge when we're talking about the state of the science when people can't even get access to the science. The last thing I'm going to mention, I was actually surprised to find that a lot of people don't know about BioArchive. So BioArchive is a preprint server run by Cold Spring Harbor. Preprint servers are not new. The physicists have been running preprint servers forever. There's actually archive.org. What is a preprint server? Is as I'm working on my research and I have a paper that is, near finalized, what you do is you throw it on a preprint server that is open to everybody in the world, and then people can comment on it, before it goes to publication. So the BioArchive has exploded. I could show you a graph in popularity. The concern used to be, if I disclose my research before publication, then the journals are not going to accept it. This is actually, because of the popularity of BioArchive, all of the journals have had to bend over backwards to accommodate BioArchive. So there are now groups from Broad Institute, pick your, top research institute that are putting their papers in BioArchive, and these are still being accepted in Science, Nature, blah, 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 et cetera. Most major journals have changed their publishing policies to accept preprints. So you can actually read about their policies. There's a few bad journals that haven't, but we should be able to put pressure on those. So I would just encourage, since it is so hard to get access to this research, that the consortia could potentially think about embracing BioArchive. We're trying to put our money where our mouth is, and one of our early papers from the consortia efforts is actually published on BioArchive. That means that people can access the full paper, all the results, all the data, et cetera. We'll still be able to publish this paper. There's no boogeyman that's going to come and scoop us. It doesn't happen. So a lot of people have done research. So I'm out of time. I'm going to get off my pulpit here. And I'm sure there's a bunch of questions that came up from the first two speakers. All right. Thanks.